Good afternoon. I'm Liz Siegert, and I'm the Health Beat Leader on Aging for AHCJ. This webcast is one in a series of topic-specific training opportunities to brief health journalists on the latest information in a particular health area and give them story ideas or story resources they can use in their work. Today's topic is how research misconduct harms patients and science, and it's especially pertinent in light of the recent funding freezes, firings, and cuts at the National Institutes of Health. Many people are already skeptical of even basic science, so any red flags only exacerbate the mistrust we've already encountered. I'm delighted to introduce investigative reporter Charles Pillar of Science and author of Doctored: Fraud, Arrogance, and Tragedy in the Quest to Cure Alzheimer's, and Dr. Matthew Schrag, a neurologist, assistant professor at Vanderbilt University, and principal investigator at the Schrag Lab, who investigates the overlap between vascular and cognitive neurological diseases in the quest to find a cure for Alzheimer's. Dr. Schrag is with us today in a personal capacity, not on behalf of the university. We will be taking questions at the end, but feel free to use the Q&A box, not the chat. And please put your questions in there, and I'll try to get to as many as possible. So let's start off. Charles, your book was absolutely riveting and a little bit scary. For those in the audience who haven't read it yet, can you start off with a brief summary of your years-long investigation into Alzheimer's research and why you decided to undertake this effort?
Sure. So thank you very much, Liz. I appreciate being on the webinar. The work that I have done over the last few years began actually in December 2021 when I first encountered Dr. Schrag. And at the time, he had already been an expert in evaluating some images associated with Alzheimer's disease, possibly manipulated images, I should say. And he and I were talking about the possibility that I would be covering some of his work in the pages of Science magazine. And over the course of our discussion, very quickly, I learned that Matt had been able to look further beyond his initial findings that involved a drug called Somufilm from a company called Cassava Sciences, which some listeners may know recently crashed and burned and is no longer being developed for Alzheimer's disease. That's a long kind of sordid tale, but what kind of stimulated this process that led to my book, Doctored, was that Matthew found in his further examination of studies associated with Alzheimer's disease that a very important study that had been one of the most influential in the field for many years that tended to support the so-called amyloid hypothesis of the disease and was something that gave the hypothesis a great deal of renewed confidence in a period of skepticism back in 2006, that study in the esteemed journal Nature was apparently based on doctored images. And that began this years-long examination of a bigger range of Alzheimer's research by me, and I should say with incredible help from Matt Schrag and from other experts in forensic image analysis.
Well, Dr. Schrag, you became interested in Alzheimer's and neurological disorders early on in your career. For those in the audience who may not be as familiar with the history of this disease research, why has it been so difficult to find a cure or even a viable treatment? What are some of the challenges here?
Yeah, well, thank you for having me on today. I'm really enjoying being here, and to get to do one of these with Charles is a real privilege. So thanks very much. Alzheimer's disease and understanding the brain is one of the last great frontiers scientifically. You know, there's a handful of these topics that have just proven to be extremely difficult to work on. And this is one of those generational challenges that seemed like it would be a great place to invest one's life's work. When you look at healthcare development over the last century and a half, we've made incredible strides that have lengthened the average life expectancy for many, many Americans. But we're losing some of the value of those added years because of the prevalence of neurodegenerative disorders at the end of life. And so to really capture some of the benefits of all of the advances in medicine, we need to tackle this disease. And for that reason, it seemed like a good thing to focus on. But of course, it's also very personal. You can't interact with patients or their families who are suffering from these really peculiar diseases and not feel, a really deep level of empathy for them. And as a physician trying to treat them, we feel very inadequate. Our current tools just aren't up to the job. And it's not that we can't do anything for these families, because I think we offer them quite a lot, but we certainly aren't meaningfully slowing or stopping this disease. We've seen breakthroughs in other neurological diseases, but this family of neurodegenerative disorders has been incredibly stubborn.
So, research gets published all the time. And, the premise of Charles's investigation and your work as well has been focused a lot on image doctoring, which is also data manipulation, although some people may not understand that images actually are data. So can you explain kind of basically what image data manipulation is, and why you decided you would possibly risk your reputation and even your career to bring this to the attention of the proper authorities?
Yeah. Well, I remember the first time I was asked to do this in an official capacity, remarking to the person asking me if they thought I was really foolish enough to do this. And so it's sort of something that is not, routinely encouraged for junior faculty to do. When we work in a basic science environment, a lot of the experiments that we do end with a photograph. We conduct some set of analyses, either looking at the structure of a piece of tissue, a piece of brain tissue often in our context, or we run an analysis to try to understand how much of a gene or a protein is present in a sample that we've prepared. And often the final step is a photograph. And we make measurements based on those photographs. And so the actual images are an important part of the data. And it's become traditional to show examples of that data in your work. And so for this class of material, there is at least a very small portion of what's done where you can actually see and touch the source data. Which is helpful. There's huge swaths of data out there where it's very hard to get any kind of forensic analysis based on what's published. But this is one swath where you can see the source data and understand what's there. And often what tips us off that there may be problems is just looking at an image that looks funny. Right. And I think that it's surprising to people, but that's often the extent of it. Having worked in this place a long time, you get a sense of what things should look like. And when an image deviates from that pattern, you just get the impression that maybe something has changed. In the same way, if you're walking through an aisle at the grocery store and you see a magazine picture and you think, oh, maybe that looks like it's been photoshopped or airbrushed in some way. It has something unnatural appearing about it. The forensic component of what we're doing is trying to go beyond that, and trying to find ways of proving or at least strengthening the argument that the images have been altered in some way from their original point.
Well, Charles, talk a little bit about from the media perspective, something gets published in a scientific journal and it's supposedly touted as a breakthrough, and then the mainstream media might pick up on it. How do things like image doctoring really affect, what most people know about this disease?
Sure. So naturally, if any sort of data is false, then it has a profound effect on the validity and the reliability of an experiment. And the question, I think, for reporters is, how do you even know that? And if you're not an expert in these fields, even myself, and I've now been studying this for years, I don't really trust myself to assess whether a particular image is based on manipulation and therefore not reliable. And so I rely on others like Matthew Schrag, who have the forensic image analysis skills and the technical knowledge within the field. And so, for example, in the case of the images that were examined, the multitude of images examined for my book, I relied on sort of a multi-layered reporting process. And I can't emphasize enough that it's great for people like Matthew Schrag or others who have brought tips to me to suggest that there are problems in a study or a set of images. And, naturally, he's a very credible source, someone who I've relied on for years and have grown to completely trust. And actually, even at the beginning, his credibility seemed very good with me. But you have to use your normal reporting process in order to validate concerns that are raised and to understand their context in the scientific world. So for me, it's two layers. One is that I need to go to other experts in forensic image analysis and to understand whether the claims that are made by Dr. Schrag or anyone who's bringing these things to me are well-founded, well-constructed, thoughtful, and that a person could, in fact, sort of reverse engineer that process and come to the same conclusions. Once I've done that, the second step is understanding its meaningfulness in the field. There are all kinds of manipulations that can be done. Some are even a kind of misconduct that don't have a really substantial effect on the field or even on the understanding of a particular experiment. So you have to reach out to experts within that scientific field. And of course, in my situation, experts in Alzheimer's disease to understand both whether they also agree that this is important, but also what its effect might be in the understanding of what this experiment was saying and what influence it had and perhaps shouldn't have had in the field. So once I've gone through those two steps, I know that I can do my further reporting to provide an analysis for readers that puts it into that bigger context.
A lot of the investigation that Dr. Schrag looked at and what you reported on, Charles, had to do with the amyloid plaque theory. I think most of us have at least heard about this, but I'm wondering, Dr. Schrag, if you could kind of, explain it in a couple of sentences, if that's even possible.
Sure.
And then both of you maybe talk about how that's kind of harmed a look at perhaps other theories of treating or curing Alzheimer's.
Absolutely. So over about 120 years ago, when Dr. Alzheimer's first discovered these types of changes in the brain, one of the things that he remarked upon was that there's the formation of plaques in the brain in patients who have the disease that would become named after him. And they also have tangles within neurons. So there were these two distinctive changes. And the plaques, which form outside of the cells between the neurons, essentially, over time became recognized as a potentially toxic driver of Alzheimer's disease. And in the late 80s and early 90s, this crystallized into what became known as the amyloid cascade hypothesis, which put the clumps of this little tiny protein amyloid at the very front of the cascade of events that leads to neurodegeneration, and argued that therapeutically we should be targeting this material either to prevent it from accumulating or to try to remove it from the brain as a therapeutic strategy. And this idea has been the dominant idea in the field for 30 plus years now. It has driven the majority of the large scale trials for disease modifying therapeutics.
So how does this impact, and Charles, you touch on this quite a lot in the book too. How does it impact scientists who are not, embracing this theory 100%, that maybe want to look at other approaches to, research and treatment?
Sure. So I talk in the book about a sort of term of art, the amyloid mafia. And I want to make sure people understand this is not a real organization, but it references a way of thinking in the field that became so dominant that it tended to crowd out other ideas. And it took various forms. For example, when a scientific idea achieves such extreme prominence and mind share, it can be one that results in skepticism towards alternatives to that idea. And this is exactly what happened in Alzheimer's disease. It's not that there aren't a lot of other ideas about what might be the cause of Alzheimer's and what therapeutic interventions might be beneficial, because there are many. It's that there tended to be a reluctance to fund those adequately to result in progress that would either prove their possible beneficial influence on the field or disprove that. And because of that, there's a lot of ideas hanging out there that just have been orphaned in a way. The other thing is that there has been pressure from leading figures in the field to emphasize the amyloid hypothesis approach, not just above others, but to the exclusion of others. And you see that, and when Matthew Schrag mentioned the issue of major trials, most of the big money in the field, the big money, when I talk about top 100 pharma companies that are involved in financing gigantic amounts of funding for large-scale trials to try to test out drugs in the field. These are primarily oriented towards the amyloid hypothesis. And when you hear critics of the idea that, people who push back on this idea that there's this dominance, um, they often say, well, there are many other ideas that are being explored and many other trials that are going on. These are very often, most often, very small scale earlier trials that don't involve huge expenditures that you see with the amyloid related trials. So I guess I'm just saying that on multiple levels, this dominance has been exerted in a way that has marginalized other ideas, that has actually caused scientists in the field to change their ideas to concentrate on amyloid related therapies or research.
So what, now that a lot of this information has become public, both through Matthew's efforts and your efforts, Charles, what's been the reaction of neuroscientists and academics in the field? Have you gotten a lot of pushback, or are people starting to come out and say, yeah, I always thought such and such's research was a little flaky?
Yeah, it's an interesting perspective. There has been a little bit of pushback, but I think there's also been a fair amount of support, at least for major components of this. I think that the troops on the ground almost all have a story of encountering problematic data at some point that threw them down a rabbit trail. I'm amazed when I give talks and when I interact with people, how many people pull me aside and say, I have a story. To the point that, I think that this is something that we as a profession recognize on an individual level. And it's interesting to start hearing the stories on a larger scale to realize that research integrity may be a bigger problem than we thought it was. And that's not to say that it's everywhere or that it's anywhere near the majority of the science is being produced, but I think it's enough. And sometimes it can occur at strategically important nodes in our scientific thinking. That means that we're probably all going to interact with this problem. And I think that there has been a recognition, especially I think with younger scientists who are really engaged with the fine details of the work, that this is a problem. I think the implications for the specifics of the amyloid cascade hypothesis have been a little bit more controversial, and I think there's room for different interpretations there. But I think a lot of people working in the trenches also feel like we need to move past this idea. The amyloid cascade hypothesis has been effectively disproven by not just this research integrity work, but more importantly, the chain of clinical trials that have thoroughly tested this idea. And to be frank, I don't think that the concept ever made a great deal of sense for an age-related disease anyway. I can't think of another age-related disease that has a single solitary cause where there's one domino at the front of a cascade that causes the problem. That's just not how we would expect this complex organ to behave. We would expect it to be a much more multifactorial, complex process, and I think beta amyloid is one component of that. I think even among the continuing proponents of the amyloid cascade hypothesis, you're starting to hear that recognition that people are saying amyloid is a part of the story, but it's not the whole story, and we need to be focusing on other components. And so while I think there's been some discussion, I think there is a general recognition that we need to move to the next generation of Alzheimer's research.
Charles, I'm wondering too, as this information has come out, you know, there are so many people out there, we've all dealt with them, who are basically skeptics of almost anything to do with evidence and science. How is this fueling, you know, that side of the argument, saying that, you know, all of science is riddled with fraud and you can't believe anything and so on? How do journalists, you know, how do we kind of counter that?
It's a big problem. So as someone whose work has occasionally been used to bludgeon science more generally, of course, it's something that is very disturbing. Um, let me just say that one thing that we have to do, and something that I do in every interview I give about my book and in every opportunity, is to state quite clearly that, like every human endeavor, there is some misconduct and some fraud in science generally and in Alzheimer's research specifically. And uh, this is just part of the human experience. And we would, just as we would never say that all plumbers are corrupt and you should never hire a plumber to fix your pipes, we really don't want to jump to any conclusions about the corruption of science more generally, because we've identified a few people who have engaged in misconduct. Let me, if I may, just take it one step further to broaden it to this discussion about why we're here and what might be possible remedies for understanding better how to attack this kind of misconduct in the field. So, in my book, I look at the institutional authorities of science. And when I say that, what I mean are the funders like the National Institutes of Health, the regulators like the Food and Drug Administration, universities, and journals, which are both kind of the gatekeepers of scientific knowledge and have the responsibility for overseeing the process of disseminating knowledge in such a way that it's reliable and that it's well policed. Well policed for, again, these relatively rare examples of misconduct that could nonetheless be influential and harmful. And what I've seen repeatedly is that all four of these institutions that we depend on for advancing knowledge and providing eventual cures for dreaded disease, they've been complacent and they have been remiss and they have been, I would say, arrogant in their approach to understanding how to evaluate possible misconduct and how to push back against it and to examine it and to do things that make it less likely to have influence in the field. And so right now, of course, we're in this situation where there are a lot of attacks on science, not just by individuals who are very, very skeptical and who, quote unquote, do their own research on the internet, but also by government institutions now in the Trump administration who are potentially using a kind of slash and burn approach to funders and to regulators in a way that I think is horrible and disapprove of. But I think in a way we have to be clear that being critical of the performance of some of these organizations, calling out their complacency and their lack of leadership and the places where they just simply have to up their game. This is essential to people who care about advancing science. And for me as a journalist, if I tried to ignore those, as I think some scientists are saying, well, we can't bring out these issues because it gives fodder to the critics of science and it's kind of, in a sense, anti-science. I think this is incredibly wrongheaded. And they're essentially saying to us as journalists, don't be journalists because science is under attack. This is a philosophy that is so transparently wrong and so harmful in the long run to science that we have to resist it.
Yeah, I mean, you brought up about 3 or 4 things I definitely want to follow up on. But first, I think, you mentioned the Trump administration and what they're doing, and we have to address the baby elephant in the room, which is, the funding freezes for Alzheimer's research. They've just closed, unless they've rehired them, I can't keep track anymore, of the Alzheimer's Disease Research Center. Scientists who are in the middle of investigations are scared to death that if they use certain language in their grants, that their funding might get cut off. I'd like both of you actually to address what this is going to mean for the future of research if these funding cuts and freezes and closures and everything else that's going on in this tornado is allowed to continue. Matt, you want to start?
Well, I'll start by saying that we certainly don't know what's going to happen. But what we do know is that this work is critically important, that there are millions and millions of patients who are depending on us to develop treatments for this disease. So one way or the other, we're absolutely committed to continue moving forward with the work that we're doing. And I think that's the critical piece. The other thing that we as scientists need to do is that regardless of the headwinds, we need to maintain our standards at a very high level, right? It doesn't, what we're looking for when we're looking to build public trust is not for people to trust in some sort of propaganda or some sort of slogan, but really for us to be trustworthy and have people trust us because we're doing the work and we're honestly holding ourselves to a very high standard. And so I think, I've been telling people for years now that in season and out of season, the message internally to our profession is the same, that we have to have a very high standard of rigor. We have to have a very high standard of integrity. And Charlie mentioned earlier that there's a certain amount of fraud that is just going to be an intrinsic part of a complex human endeavor like this. But what doesn't have to happen is that as a scientific institution, as a scientific profession, we don't have to be naive to that. We can create structures that are better able to identify and deal with those types of problems, that are a little bit more skeptical in terms of how to respond to breakthroughs before they're replicated, and make specific investments in some of the boring aspects of science that are critical for making sure we're making forward progress. And so I think that the current political environment, or any other political environment, is interesting, and it comes with its specific problems. But the message from within our profession should be much more durable and much more consistent. And that's that we maintain high standards and we try to build a real trust with the patients and the population that we serve.
Charlie, you want to add on to that? I'm also curious because would the current environment maybe prompt some scientists, not all scientists, to maybe fudge their data a little bit or skew the results a little bit more to kind of appease the powers that be to ensure funding? I mean, it's almost like a vicious cycle.
Yeah, my view is that I don't think you'll see any more of that than we already see, which is that that's a human phenomenon. A small percentage of scientists, just like, as I mentioned, other walks of life, are going to cut corners and engage in activities that are either corrupt or improper in order to have a leg up in a very competitive world, a very competitive world of science. I think, part of the problem that we're facing here is that people in the public are, they're being told certain things that they don't see the evidence of with their own eyes. Alzheimer's is a good example of that. And the place where I wonder about the message being sent by some of the leaders in Alzheimer's disease is they're saying we're on the right path. Particularly with the amyloid hypothesis, they're saying we have been treading this path because we know it's correct. And there are many who, unlike what Matt just said, many who have already declared victory and said, we know we're right, we're on the right side of history, is what one scientist said. And they're going to continue down that path regardless of what the results are. Patients and their families are looking around and kind of getting like the old joke, are you going to believe me or your own lying eyes? They don't see that progress in a way that is materially effective for them. And so what I think about the current situation is that the phenomenon of the Trump administration and some of the press for really radically changing some things in the government is, I think, to some degree, a response to the complacency and the arrogance that some of those institutions have at times shown. And uh, while I completely disagree with the approach being taken, I think it's extremely harmful and really should be resisted. Um, I do see that we should be pushing those institutions for reform and for doing better, doing better at their jobs. Let me, if I may, Liz, I can give one example of
Sure.
something that I think captures this in a way that people have a right to demand better performance. So this has to do with a investigation that appeared in Science that I wrote with the great assistance of Matt and other forensic image experts who worked for a very long time in compiling information from the research, 25 years of research of a guy by the name of Eliezer Maslia. He used to be the director of the Division of Neuroscience at the National Institute on Aging. And the reporters on this call may recall that the National Institute on Aging is, and in particular that division, are at the pinnacle of decision making on what gets funded and what emphasis there is in the field of exploring neurodegenerative diseases such as Alzheimer's and Parkinson's disease. This guy was sitting in the chair that had enormous influence. Now, the investigation that appeared in Science showed that he had, for 25 years, been apparently doctoring images in, or I should say, there were doctored images in his work going back 25 years, more than 100 such studies, including the reuse of images improperly across studies in different years, different publications. These are really egregious concerns. And so just to cut to the chase, after my story appeared, the NIH revealed that it had found misconduct in 2 studies by this guy, 2 studies out of 132 that were identified in my story, and that he was no longer going to be the head of that division. So this is a good outcome. They made their announcement on the day that my story appeared in Science last fall. But the most telling part of this, the part to me that I found the most shocking in a way, even as shocking as this whole story might seem, is that when I asked the NIH, do you look at your high-level hires' scientific history for the possibility that they had engaged in image-related misconduct before you make the hire, or will you do that in the future? The answer was no, we don't, and we won't, because we don't think it would help the research at our institution. And to me, this was an example of the arrogance and complacency that we're talking about. So when you look at what the Trump administration's doing, when you look at the slash and burn approach, I say no, that is extremely wrong-headed. If they go through with this, it'll be incredibly harmful to the field. But when you ask, should these institutions be asked to re-examine their practices? Should they be required, demanded to do better, to be more vigilant and be more careful and be better at policing the field? Of course they should be.
Great, thanks. I want to turn to Dr. Schrag and kind of roll 2 questions up into one. Going back to the image manipulation that Charles just referred again, even to the top people at NIH. One of the questions is if you could describe, some of the things that were actually manipulated, and then maybe talk a little bit about what you're doing in your own lab to try and, because you mentioned this before, some alternative hypotheses or building onto the amyloid issue.
So we see a pretty wide range of potentially problematic data in the forensic work that we're doing, but specifically in the image space. you can see very simple problems like an image appearing twice representing two different conditions. That would be a very common type of problem. And these are the sort of things that can happen simply as an error. If you're building complex panels of images with lots and lots of different components to them, you could imagine grabbing the wrong file, especially if they were similar, related conditions, and inserting the wrong image in the wrong place. And so something like that we would often detect. But the thing that really raises eyebrows is when we're seeing are anomalies in the data that are a little bit more complex. So the more complexity to the change, the more that it looks like the change has been disguised or done in an intentional way, the more likely it is to be a problem. And so sometimes we see within an image that there are areas of cloning where components of an image may appear multiple times, either within a single image or across a number of images. That's something we saw with a considerable frequency in Dr. Maslia's work, and we've seen in other scientists that we've studied as well. And that takes a degree of intentionality to go and clone components of the image. But even then, it's hard to know for sure why that's being done. And so, sometimes these types of changes, even though they're clearly inappropriate, could be done for purely cosmetic reasons. Perhaps there was a blotch on the image or something that made it unattractive or distracting in some way. And so you ultimately want to use these red flags as an argument to ask for the source images and compare what we're seeing to the original data. Because when we're looking for fraud or when we're looking for true manipulation, we need to show that there's been a change to the image, but also that that change is intentional and directional. Right? We're not talking about mistakes. Mistakes happen. This is a complex business that we're in. And I think in anybody who's got a substantial body of work, we're probably going to find some mistakes. And that's expected, right? When we structure studies, we're trying to control for error. And the error is not just in our instruments and in the animals or cell cultures or the tools that we're studying. Some of the error is in the people. And so we structure our studies to try to be resilient to that error. What's so problematic about true manipulation is that it's directional. Error tends to be random and should come out by using rigorous techniques. But when you have this pervasive, directional, consistent types of changes across a body of work is when we see alarm.
So how have you taken some of this into account in your own research? If you could talk a little bit about what you're working on now.
Yeah. So I think when we look at what might have been crowded out by the amyloid discade hypothesis, you worry that we may not even know necessarily the answer to that question. When researchers decide what to study, one of the first things that is critical is to keep the lights on. And so when you invest too much of the funding after one idea, you don't create a competitive marketplace for these ideas to bounce off of each other, and you may lose some depth in the field. Our laboratory is focused on understanding the contribution of blood vessels to Alzheimer's disease. Most people don't think of Alzheimer's disease as a blood vessel disease, but it absolutely is. And a substantial percentage of patients who have this disease have deterioration of the integrity of the capillary network and the little tiny arteries that feed the brain. And we think that's a major contributor to cognitive symptoms. And it also sets up some of the side effects for the existing treatments. So when patients who are treated with these new antibody drugs to remove beta amyloid, a certain percentage of them develop a side effect called ARIA, which appears to be a breakdown of the blood-brain barrier related to blood vessel injury. And so our laboratory has focused on trying to understand that and hopefully come up with a class of therapeutics where there's synergy between the changes that are happening with neurons and in the brain tissue and in the blood vessels. There's also a great deal of emphasis now on trying to understand inflammation. Smoldering low levels of inflammation in the brain may be really important, and that could be driven by any number of things from past traumatic injuries to the brain or strokes or even infections of the brain at some point. But trying to understand the long-term effects of low-lying inflammation seems like a promising area, and there's at least a half a dozen other ideas that I think are worthy of taking center stage in this disease.
Wow. And, it's the idea of getting this type of research published to kind of open up people's vision of what could be. Have you run into issues with, well, it's not directly amyloid, so maybe, you shouldn't
Yeah.
focus on it?
Well, often it's not quite that direct, but occasionally, I recently had a grant review where that was one of the concerns, was that we were too critical of the amyloid cascade hypothesis, and so the work shouldn't be funded. And that occasionally comes up that directly. But I think more often than not, it is a quieter sort of discrimination. But I will say that one of the examples that Charlie and I explored in the work that he ended up publishing in the book was looking at how peer review processes in one particular journal, although we see echoes of this in other places, could be altered to favor certain ideas and certain scientists, where papers that were published with editors as co-authors, or who had the right ideas or originated from the right sorts of companies, were given priority in terms of how rapidly they were peer reviewed, or perhaps whether they were peer reviewed at all. And so I think that part of what we would argue is that if you compromise other aspects of this process, if you make peer review less rigorous, you open the door for problems like research integrity lapses to have a bigger effect.
Charlie, one of the things you mentioned in your book, and I'll take you back to 2006 with Sylvain Lesnay at University of Minnesota. Apparently, this young researcher has been fooling around with a little bit of images and data for quite some time. So this problem is not new. Could you kind of recap that story very briefly and talk about the percentage or the propensity for this kind of issue?
So Sylvain Lesnay, he was a postdoc in 2006 in the lab of an illustrious Alzheimer's scientist by the name of Karen Ash, who had made other very important contributions to the research field. And Lesnay, he's a French native and had been integrally involved in the development of these images for this important 2006 paper that was published in Nature. And of course, people, I'm sure, know on this webinar that Nature is one of the most important scientific journals in the world. So when you publish there, you have a lot of influence usually. And what was particularly interesting is that in 2006, there had been increasing skepticism towards the amyloid hypothesis that had been building. And as Matt mentioned, there had already been therapies developed that were very good at removing amyloid plaques from the brain, but very ineffective at arresting or reversing the symptoms of Alzheimer's disease. So there was some discouragement about it. And when this experiment was done, it was a very clever experiment where essentially they were working with genetically engineered mice that produced copious volumes of amyloid proteins in their brains. And they extracted these proteins and refined one specific type of amyloid protein called amyloid beta star 56. So their star protein, this is what goes for a catchy name in the scientific world. And this immediately grabbed a lot of mind share because the conclusion of the experiment was that they injected this protein into rats. And very quickly, those rats began to show signs of memory loss that were described as being akin to what people experience with Alzheimer's disease. So you had, for the first time, a kind of cause and effect relationship between a particular substance and the symptoms of Alzheimer's disease. And so this experiment immediately grabbed an immediate kind of dramatic mind share within the scientific community. It received enormous attention and became one of the most cited laboratory experiments for the next 16 years in the field. And it really reinvigorated and reinforced the idea that people who were committed to studying and proving out the amyloid hypothesis were on the right path. And that's why when Matt found these apparently doctored images within this seminal experiment, and when I was able through my reporting to validate his findings in multiple ways, including with one of the most foremost advocates of the amyloid hypothesis, that you saw that it was something that really called into question one of the bedrocks of that set of ideas. Now, I want to make a small observation, if I may digress slightly, Liz, in telling a little extra bit of this story. What happened after my story about this appeared in Science in July 2022 is that you had an immediate response from the major scholars in the world of Alzheimer's research. And they started to post their comments about it in this website called Alzforum that people may be familiar with, kind of a watering hole for Alzheimer's scientists and a compendium of research there. And the responses broke down in two ways. One was, this is a moment when the field could use it as an opportunity to take a good hard look in the mirror and ask themselves, have we been essentially putting too much stock in something that should be treated as perhaps important, but not everything there is to know about this disease? In other words, amyloid proteins have something to do with Alzheimer's, but not everything to do with it. And should we be reorienting towards other ways of looking at the disease and broadening our horizons? The other set of responses were we knew all along this experiment was suspect. We never gave it that much credence. But if you look at the people saying that second kind of response, for years, over and over and over again, in their own scientific papers, they were citing this experiment as a way of supporting their own ideas in the amyloid hypothesis and to proving that their ideas were on solid ground. So I would call this rank hypocrisy. And so what you have is a kind of set of conditions where you see so many scientists are having trouble facing up to problems in the field that I think a more forthright examination could really benefit everyone and move the field forward.
Do you have any sense, you mentioned it's only a small percentage of, scientists or researchers that, you found between the two of you that really are not playing, along a straight path here. Do you have any sense, is it 1%, 2%?
Well, I mean, there have been studies done of this, and you have to understand that it's really impossible to get a good answer to that question. But I would say between 1% and 2% is a generally accepted idea about the amount of both image manipulation and other forms of apparent misconduct in scientific papers. But let me just emphasize something that Matt said earlier that I think is very important to keep in mind. It's not so much the quantity of this, because we know that the vast majority of research is honestly done. What it is whether certain falsified experiments, if strategically placed in a set of ideas that's influential, can skew thinking in the field improperly and lead people to conclusions that waste money, waste time, and squanders opportunities to find benefits for patients.
I really want to emphasize this point because I think that it's not just the total amount of research misconduct, but also the way that as a field, as a profession, we deal with those types of problems that really allows them to have a disproportionate impact. When these types of problems are identified, the interactions with the various authorities who are responsible for addressing this and people like myself who bring these issues to light is often quite hostile. There is this sense that the profession doesn't want to hear about this. When there are formal investigations into how these things happened and whether or not research misconduct has taken place, almost all of the authority is delegated to the host institution, to the institution of the individual whose work has been impugned. And so there is an intrinsic conflict of interest between the people who are tasked with investigating these concerns and the accused parties, because they share research dollars, among other things. They also share reputation and they benefit from the publication. So there is this this lethargy in the field to address this because there's very little to gain by outing these types of problems. And pretty frequently you'll hear from leaders in the field, we don't need to take this type of approach because science is somehow self-correcting, and that if we can't reproduce things, people will simply move on. But that process can be exceedingly slow, can lead to a lot of work going down rabbit trails, and it can leave problematic people and pipelines of data in place for a prolonged period of time. And so some of these issues that we raised, 4 years ago, and some of our colleagues have raised issues over a decade ago, are still on the books at these journals, even though they are overtly problematic pieces of data. And so for all of the people who criticize this work by saying that we're painting with too broad of a brush and that these very unusual actors in this field are not compromising the whole field, the actual, I think, most problematic elements are not the individual fraudulent actors, but how the field writ large is responding to them. And the fact that these individuals have on occasion gotten to positions of extraordinary power speaks to the lack of the ability of the field to detect and hold accountable people who are doing these sorts of things.
So self-policing isn't really the best option here.
Well, no kidding.
Charles, how do we as journalists kind of push back on this? I mean, you were fortunate that you had the support of a major publication to do this investigation. You had years to work on it. Many of us don't have that kind of luxury. So what can journalists do if they think, they spot a study that may raise a few eyebrows or think that it's, maybe not 100% above board?
Sure. I am very privileged to have time to work on these larger issues, larger stories. And I would just say that not every examination of research has to be something that takes months or even years to do. I would suggest that people use a couple guidelines. One is understand whether the concern being raised is important or not. Know your field well enough to ask yourself this question. Even if the worst is true, even if this complaint is really true and the study is invalid, is it important enough to spend a bunch of time on? Second, arm yourself with consultants who are able to help you assess both the truthfulness of the concerns about possible image manipulation or other data manipulation, and also the meaningfulness. In other words, how best to answer that question of importance. If you're not yourself a true expert in a particular field, find people that are, who can give their feedback. Ultimately, you're going to need to interview those people anyway. And I would say also, something that is so important to me is approaching these kinds of stories, small or large, with a sense of seriousness of purpose that reflects how it might be very damaging to the subject of your story or your investigation. These are scientists who have, many of them, labored their whole careers to get where they are. And if there are questions about a study that actually can be shown to be well demonstrated, they can destroy careers. And I personally have been involved in stories that have resulted in radical changes in people's lives and careers. And I don't take any pleasure in that, but I do take satisfaction in exposing wrongdoing that should not be allowed to continue. But one of the things that I always say and do in these stories, small or large, is there's no surprises. When I am writing a story like this, the subject of the investigation knows everything I know. They have a complete set of information. Sometimes this is a lengthy dossier on their work. Other times it's shorter. But not only that, I provide them with a list of questions that reflects the issues that I'm planning to cover in the story. Pardon me. And the reason I do that is not just for fairness to that person, to give them every opportunity to tell their side of the story, but it's for the reader. It's because the subject of these stories, they may know information that no one else knows that's essential to providing the best, most informed look. And so I always take it seriously as a reporter that this is central to my job. Now, I find most often that the subjects of these investigations don't respond because they don't have a good story to tell. I think it's often a mistake. It's almost always beneficial to them to tell their side of the story, even if it's an equivocal kind of response. But I think this reflects that there is unfortunately, a lot of misconduct that people are trying to hide.
So that's a good segue into our next question here, which is what can society do to ensure that research is legitimate and that penalties are placed against those that violate the public trust? Matthew, you want to take that one?
I think that we're not going to make fraud in this space go away. I just don't think that that's realistic, but we can work to build structure into what we're doing that makes it more resilient. And I think that taking these issues seriously and making sure that there are real consequences when these issues arrive can dissuade some people from taking these kinds of liberties with the data. So I think those are all important things. I think that as someone who runs a laboratory and supervises quite a number of people, I think that the level with which we engage with our own data and with our own staff and trainees needs to be pretty deep. I think that people who are leading labs need to be plugged in. And some of this is actually structural, right? I think that some of the mechanics of how we do research would benefit from being a little bit less trusting. I think that using old-fashioned pen and paper lab books to keep data is clearly an opportunity for fraud. I think siloing information so that a relatively small number of people have access to the raw data is also a very common mistake. And so I think that as we move into the next decade of research, we should be thinking about what should change with sort of the routine day-to-day of how data is produced and managed to make the system a little bit more resilient to this problem and to help those of us who are running laboratories identify when data has changed so that we can ask why and make sure that we are deeply engaging with our own data. So I think that's a really important component to this.
Well, we only have about a minute or two left. Charles, any final thoughts? People have questions. How can they reach you?
Sure. So you can find all my contact information on my website. That's charlespiller.com and all one word.com. And I would just say that I want to thank Matt for his incredible courage in bringing forward these issues. It's much easier for me as a journalist to write about this. This is my job. But for a scientist to do this and to challenge the institutional authorities of his field, that takes a lot of bravery because the potential consequences can be severe. I'd like to say the same thing about a lot of other scholars and forensic image analysts out there who have taken personal risks and also committed themselves to really bettering scientific institutions, because that's what this is all about. It's about trying to create a culture in a way of administering science that's more reliable and more beneficial for all of us.
Well, unfortunately, that's all the time we have today. I'd like to thank both of you for the incredible work you've done on this issue. And Matthew, do you mind sharing your email with our audience?
Absolutely. My email is Matthew, MATTHEW, .schrag, SCHRAG, at VUMC.org. I'm more than happy to hear from people.
Great, thank you. And thanks to everybody who tuned in today. If you joined us late, you'll find the recording of this webcast at healthjournalism.org in a few hours. Once again, thank you to our presenters for sharing their expertise. AHCJ will continue to bring you webinars on a variety of health topics as a perk of your membership. And good luck to everybody on their stories. Thanks again to both Charles and Dr. Matthew Schrag.